Well, now that we've seen how the gene was discovered, let's look in part 2 of how the gene works and how knowledge of the gene can help unlock more of life's secrets today and benefit all life on Earth, including human beings.
Alice laughed. "There's no use trying," she said. "One cannot believe impossible things." "I dare say you haven't had much practice," said the Queen. "When I was your age, I always did it for half an hour a day. Why, sometimes I've believed in as many as 6 impossible things before breakfast."
And if Alice could wander through the wonderland of modern genetics, she would need even more practice. For here at the very heart of life, the impossible is an everyday thing.
Right now, today, we can transfer a gene from one plant to another and from one animal to another. We can make a synthetic gene for rabbit hemoglobin. We can create new kinds of living bacteria. And some of these bacteria can mine copper for us, can make insulin and other hormones, can produce new antibiotics, can fix nitrogen into fertilizers, and someday soon may even be able to produce and reproduce living computers.
Researchers have now been able to make exact duplicates, clones that is, of sheep, pigs, mice, cows, and many other common mammals. No one has yet cloned a human being, though this is probably possible. In fact, if we look only at the possible, less than a teaspoon of a chemical called DNA, deoxyribonucleic acid, would be enough to recreate all the people now living in this world.
Now, all of this is not science fiction. Scores of hard-nosed business people have invested millions of dollars in new high-tech companies springing up all over the world to make these impossibles everyday realities.
So let's look at the basic science that makes all this possible. Just as knowledge of the atom is the most important key to understanding physics and chemistry today, well, knowledge of the gene is the most important key to understanding biology today. So what is a gene?
Gene, we know today, is a molecular structure that carries the code needed to govern life functions. Just as a computer needs a program, so a cell and a complex combination of cells called an organism needs a program. That program is in code, and the code is on a spiraling molecular structure called deoxyribonucleic acid, DNA for short. A molecule of DNA actually carries more than one instruction, more than one gene. Along this spiraling DNA molecule, for instance, one section might be part of the code that, in interaction with other genes, governs eye color. Another section could have a code needed to produce the hormone insulin. Another section would have a code for hemoglobin in your red blood cells.
Obviously, in order to create an entire person, you need a lot of genes. How many is an active research question today. It used to be thought humans had 100,000 or more genes. Well, the most recent estimate is much smaller, probably on the order of 25 to 30,000. With this relatively small number of genes, not actually that many more than a fruit fly, has complex interactions that multiply into billions. In fact, the possible combinations, of course, get astronomical. How else could we have such a rich variety of living things in our world?
When you think of all the possible combinations of magnetic pulses on a tape, grooves on a record, words in a book, DNA is like that, only more so. More information and more power. Now, the DNA is surrounded by protective protein coats and is strung like beads along structures called chromosomes. These chromosomes wind around in every cell's nucleus. The DNA in each chromosome has thousands of genes. One surprise is that much of the DNA does not seem to carry any coded messages at all.
We don't know at this time why this should be so. Perhaps nature, like human beings, can be redundant and wasteful, as well as efficient and powerful.
Just about every cell in the living body of every organism has a store of DNA in its nucleus. The same store as in every other cell of the same individual organism, and that's why cloning is possible. Cloning has been known and practiced for many years by gardeners, but recently it has been done experimentally with animals too, including mammals. Cloning is a way to use the coded instructions of a general body cell to reproduce the whole organism without sexual recombination. Thus, the cloned individual turns out to be exactly like the parent in every respect, since he or she has exactly the same genetic instructions within each and every cell.
Normally, of course, this is not the way organisms reproduce. Normally, the genes that are going to be used to create new organisms are very early along segregated from other body cells.
They're kept separate in the ovaries of the female, the eggs, and in the testes of the male, the sperms. Because they are segregated so early, experience of the adult organism has nothing to do with the heredity given to the offspring. Thus, inheritance of acquired characteristics is not possible. When it comes time to reproduce, cells in the testes and in the ovaries go through a special kind of division called meiosis. In the diagram, you see a microscopic view of what happens to the chromosomes in meiosis. They divide in two. Each sperm and each egg thus gets 1/2 the regular number of chromosomes and genes. In your case, for instance, this means you got 1/2 your inheritance from your mother and 1/2 from your father. When your parents had sexual intercourse, these half-number cells joined together to make a new whole-number cell, a fertilized egg. The egg from your mother and the sperm from your father, in other words, fused together to make one complete cell, a fertilized egg cell, that in a few short months was born as a unique new person.
You. In those few short months came the impossible part. The part we still know so little about it seems near miraculous.
The single fertilized egg cell had all the codes needed to create you.
To let these codes do their job, all the fertilized egg cell needed was the right environment. Your mother's uterus provided that environment. It provided the right temperature and the right chemicals.
It provided a salt-watered world very similar to the ancient oceans, where amino acids, sugars, salts, and fatty acids are abundant and ready for use. When all is right, the process unfolds like the most dramatic motion picture you have ever seen. The single fertilized egg cell quivers, doubles its chromosomes, and divides into two. This is called mitosis. Notice it is different from meiosis in that the chromosomes, and thus the DNA and the genes, doubled before the cell divided into two. Thus, each new cell has a full set of chromosomes, DNA, and genes. Let's look at this doubling more closely. Remember how Crick and Watson put it in their epoch-making article? It has not escaped our attention that this double helix structure provides powerful hints as to methods of replication within the cell. Well, here's what they meant, and here's how it does work. The double backbone of the helix is made of phosphate sugar groups. These are connected, like rungs on a twisted ladder, by 4 different kinds of chemical bases: guanine, thymine, cytosine, and adenine.
Well, guanine, it turns out, can only hook itself to cytosine, and adenine can only hook to thymine. That is sufficient, though, to create an intricate code and to provide an ingenious way of replicating.
With the help of other important chemical molecules called enzymes, the 2 backbone parts of the DNA molecule untwist, and then the whole molecule unzips. This gives 2 half DNA molecules, but 2 halves with highly active base parts.
See, now here's where the environment comes in again. Floating in that saltwater soup are plenty of guanine, cytosine, adenine, and thymine groups.
Where'd they come from? Well, from the food that the mother ate and supplied to the uterine environment. With the help of still other important enzymes, each half of DNA hooks onto the base group as needed. That is, every guanine hooks onto a cytosine, every adenine to a thymine, every cytosine to a guanine, and every thymine to an adenine. Phosphate sugars are also nearby, and so we zip back up, and now we have 2 DNAs, where there was only one before, and 2 identical DNAs. The cell now divides, the chromosomes divide, the DNAs divide, and you have 2 complete cells with 2 complete sets of code. Each cell identical to the one it came from. And the process continues. 2 become 4, 4 become 8, 8 becomes 16, 16 32, and so forth and so forth and so forth, and we get millions and billions of cells, all with the complete set of chromosomes, DNAs, and codes needed to make you, you. But wait, how come the cells are not all alike then? The cells in your eye, for example, are not like the cells in your ear. The cells in your lungs are not like the cells in your biceps. How did they all become different? For the most part, that is still a great mystery. No one knows. Not yet.
This great mystery is given a name. It's called differentiation. Now, there's a lot we don't know about differentiation. We do know, though, that it's governed by the same genetic code that's on the DNA molecules. And we know a little of how the code is transmitted from DNA in the nucleus to the rest of the cell.
The DNA is able to pass along its coded messages to a very similar single-chain molecule called ribonucleic acid, RNA. This smaller RNA molecule is able to slip out of the nucleus and into the rest of the cell. And there it takes charge of the building of new enzymes and proteins. And finally, it is these enzymes and proteins that do the actual work of the cell. Carrying oxygen, for instance, if you happen to be a red blood cell. Carrying electrochemical pulses, if you happen to be a nerve cell. Contracting, if you happen to be a muscle cell.
Amazing. Amazing not only that it works, but even more amazing that scientists have been able to figure out how it works. Of course, it's one thing to do this in theory and another to do it in practice.
Today, there are many genetics laboratories all over the world cooperating and competing to push forward our basic knowledge of the gene. The tools of all these scientists are similar. Some of the most basic are as old and common as any in science. Test tubes, beakers, sterile culture dishes, pipettes, colored tapes, a variety of chemical reagents, Bunsen burners, and refrigerators. Others are newer and more sophisticated. Electron microscopes, electrophoresis setups, scintillation counters, ultra-high-speed centrifuges, and, of course, ultra-fast computers. The techniques worked out to probe these submicroscopic genes are ingenious, to say the least. For instance, suppose you want to cut a strand of DNA at a particular spot, say where the gene for eye color is located. No problem. Today there is a large variety of chemicals called restriction enzymes that can act as sharp and ever so accurate chemical scissors. You can buy these restriction enzymes off the shelf. Other chemicals, usually radioactive tracers, are used to mark the points of cutting. Then various gels can be used in combination with electrical charges to separate the DNA fragments.
Once separated on a piece of paper or a gel, a computer is brought in to help interpret the patterns found. And the final result is a photographic record of the sequences of guanine, thymine, cytosine, and adenine.
Whether on film or reprinted on paper, the sequences are very long. Geneticists today are a little like a child just beginning to read. They're still at the primer level. What will it be like when we reach the length and sophistication of a War and Peace length novel?
Okay, it's time to summarize the basic facts before we take a look at the controversial future, the engineering of new life forms.
1, the gene is the biological unit that carries heredity in every cell of every living organism, from viruses to fruit flies, from pea plants to people. 2, the gene itself is a molecular structure, a group of atoms that are part of a large molecule called deoxyribonucleic acid, DNA. 3. It is the pattern of base sequences on the DNA that is the code, which when translated by living cells make a virus, a fruit fly, a pea plant, and a human. Not only in general, but a particular virus, a particular fruit fly, a particular pea plant, a particular human being. 4. When you change the structure of the DNA sequence, you change the code. You change the living tissue, you change the living organism. It's like changing the computer program or the words in a book. You end up with a new program, a new book. Maybe better, maybe worse. In the case of genes in nature, the new living organism that results from a change in the gene, it's called a mutation, is usually worse off than its parent. 5. Very occasionally, a mutation may lead to a new trait in the offspring that is to its advantage. That offspring will have a better chance to survive and pass on that new mutant gene to its offspring. And that's the way change and natural selections occur. Well, what does this all mean in practical life today? For one thing, today humans are trying to engineer genetic changes for the good. And they do this by using what is called recombinant DNA technology. Say you find a gene that is capable of some desired task, like cleaning up oil spills, producing insulin, gobbling up some nasty toxic waste, fixing nitrogen in a fertilizer, or whatever. Using one of the new restriction enzymes, you snip it out of the cell it is found in normally, and then you paste it into an opened-up cell in a fast-reproducing bacteria. You give the newly enhanced bacteria enough food and living space and let it reproduce. Within a very short time, you have billions on billions of these powerful new living things, new living factories. You purify the substances they have produced, bottle them, and sell them to people who need them. Similar techniques are also being developed to cut and paste genes from one organism to another, say from a bean plant into a corn plant, or from a bacteria into a rabbit. Here at a U.S. Department of Agriculture laboratory in Wisconsin, they are using a newly invented gene gun to transfer genes that resist mold into a barley plant. If successful, these experiments will lead to new varieties of barley that can themselves resist the mold epidemic that threatens barley crops all across the northern hemisphere today. And in one of the most dramatic breakthroughs this century, biologists in Scotland have cloned Dolly, the first mammal created from a cell taken from an adult of the same species. This feat was quickly followed by the successful cloning of 3 bull calves in DeForest, Wisconsin in the summer of 1997. Gene 1, Gene 2, and Gene 3 were all cloned from differentiated cells taken from a donor cow embryo. In cloning experiments, researchers take a nucleus from a cell in an embryo or in an adult animal. This nucleus, no matter what cell it comes from, skin, muscle, gland, contains all the genes of an organism. Well, they then take an egg cell from the same species of animal. They take out the nucleus of this egg cell and insert the nucleus of the donor animal in its place. Now they implant the new egg cell into a female surrogate mother. If all has been successful in the transfer, the single egg cell will now develop in the normal way inside the surrogate mother's uterus. And when born, the baby will be an exact duplicate of the adult sheep, cow, or potentially human, from which the original cell was taken.
One of the most promising, as well as one of the most controversial, of the new genetic advances is in what is called stem cell research. Stem cells are blank cells. They are cells that have the complete set of genes, but genes that have not as yet received instructions as to what they're supposed to do in the growing organism. Some of the most potentially useful stem cells are human embryonic cells. That is, the cells that come from the early divisions of the human fertilized egg. Researchers led by James Thompson at the University of Wisconsin succeeded a few years ago in culturing these embryonic human stem cells. These cultured human stem cells are potentially immortal. That is, they can be reproduced indefinitely in culture dishes. Well, the hope is that researchers can use these cultured stem cells to learn what are the signals that make them turn into blood cells, or liver cells, or muscle cells, or nerve cells. Once they find these signals, biologists could use this knowledge to find ways to effectively treat Parkinson's, Alzheimer's, heart disease, and many other diseases and defects of the human species. Well, all of these new technologies are helping us to understand life at its most basic molecular level. They are also challenging some of our oldest and most basic beliefs. Theory, evidence, and technology that can change basic life structures and behaviors bring up serious ethical and social problems. These issues are being studied and debated today. And now that you understand the basic principles of this new genetics, you too can take a role in this debate. For in the long run, in a democracy like ours, how this immense new power will be used is up to the most important people in the democracy, you and me.